So this is Dapeng Zhou. He's from the University of Texas MD Anderson Cancer Center, and he will be speaking on the functional reconstitution of NKT cells in HIV-infected individuals and the implications on the immunopathology of HIV-TB co-infection.
Do we have the right...
And we don't have the right talk. Okay, that's fine. It's like one of those stress tests. Here are 10 slides that aren't yours.
Well, while we're getting the next setup, Dorothy, if you take a latently infected individual and treat them with INH, do the PD-1 levels drop?
Yeah, so we're following people now, but we don't have. So we had originally looked at this cross-sectionally, people who've been treated versus not, but we didn't have the before and after on the same person. So we couldn't. Did look like the PD-1 went down, but we couldn't say for sure. So now we're enrolling people.
Okay, first I should thank the organizers. It's a great honor and it's a great opportunity to present our work in CFAR Houston. So I work in MD Anderson Cancer Center, but I am a member laboratory of the CFAR Houston. So today I will talk about a subset of innate lymphocytes, termed as NKT cells. So please do not confuse with NK cells. These are T cells, natural killer T cells. So this is a subset of T cells discovered by Dr. Taniguchi and Rikin in late 1980s. So they express the markers for T cells. However, they also express the markers for natural killer cells. So from the development point of view, they are thymic dependent T cells. Functional-wise, they are innate cells, so they are similar to natural killer cells. So another person who made a major contribution to the field is my ex-postdoctoral fellow mentor, Dr. Murdoch. So that is to find the CD1D restriction and the invariant T cell receptor of this subset of cells. And in contrast to conventional T cells, they recognize the lipid and glycolipid antigens. So this just quickly goes through the concept of innate immunity. So they are similar, functionally they are similar to NK cells, and they can get rapidly activated in first line of immune defense. So this shows that NKT cells crosstalk with other cells of the immune system through two mechanisms. So they can produce cytokines, and they can have direct cell-to-cell communication with dendritic cells. And the activated DCs can regulate the adaptive immune responses. So this slide shows the paradoxical function of NKT cells. That means in certain disease conditions, they can produce the Th1 cytokines. So the exact mechanisms are not clear yet. So there are several hypotheses that the ligand may determine the cytokine profile, or the type of antigen-presenting cells may influence the function of NKT cells. So in some other disease conditions, you may have the NKT cells produce the Th2 cytokines. So overall, it is a type of Th0 cells. In different settings, they may differentiate into either Th1 or Th2 directions. So this shows the NKT cells recognize the CD1D restricted lipid antigens. So this is the first ligand discovered for NKT cells. So it's alpha galactose linked to a ceramide structure. So it has been shown to be safe and immunologically active in humans. So it has been tested in hepatitis patients and cancer patients. So my lab has been working on the new formulation of this series of ligands. So we developed intranasal delivery, nanoparticle formulated ligands, and sublingual injection of such ligands to study their function as the adjuvant. So we recently proposed a study. We have a grant pending. It's the China-US Collaborative R1 grant. So this is based on the background that NKT cells are critical regulators of Th1 versus Th2 differentiation for the CD4 helper cells in anti-TB immune responses. And it is known that impaired Th1 CD4 helper cells in the HIV infection are responsible for the active TB disease. And our lab also generated new data recently. We found that alpha-gal this agonist for NKT cells, can treat established TB in a mouse model. And this has to be combined with the IFP. And we have tested both the intravenous route and the intranasal route. So being encouraged by these results, so we proposed this study, and we hypothesized that reduced Th1 and or increased Th2 cytokine production of NKT cells are correlated with the MTB disease and may serve as predictive markers for therapy. And we proposed the 3 M's to study the correlation between infection and to study the correlation between the treatment outcome and the NKT function, and also the correlation with the IRIS. So we already have done some preliminary studies with some funding from CFAR Houston. That was 2 or 3 years ago. We got funded by a pilot project to try to reconstitute the function of NKT cells in HIV infected patients. So in the field, it has been published by several labs that NKTs have 2 subsets in human, and they are susceptible to infection. And the heart therapy can recover the NKT cells. The IL-2 plus the heart therapy can expand the NKT cells. So our study was focused to answer the question whether the expansion of these NKTs is correlated to disease stage, and also whether the Th1, Th2 cytokine profiles are correlated with the disease stage. So we selected several groups of patients according to the viral load, and the method was to use the dendritic cell, ex vivo derived dendritic cells loaded with the alpha-galactoceramide, and use this method to expand the NKT cells ex vivo, and then we quantify the number and study the function to produce the cytokines. So this is the data to show that in 3 groups of patients, we can see the NKT cells proliferate similarly to healthy donors by this method. So we have to use the dendritic cell loaded alpha-galactoceramide. And this is the statistics to show the fold of expansion. So this is the phenotypes of the expanded cells. So this is tetramer staining plus the invariant T cell receptor staining, and they have the CD4 positive and negative subtypes. So this is the intracellular cytokine profiles of the expanded cells. So we can see they are able to produce cytokines. And medium viral load disease patients, we see higher expansion of IL-4, the Th2 cytokine. So the conclusion is that we can see 50 to 400-fold of expansion, and they produce Th1 and Th2 cytokines, and we observed higher Th2 cytokine from the medium viral load patients. So the outlook is, again, I show this slide again. We hope to study the function of NKT cells during in HIV-TB co-infected patients, and to study the correlation with the co-infection and the treatment outcome of the co-infection. So in the end, I should acknowledge our team members, and we have Dr. Arduino in the CFAR clinical core, and Dr. Cortinez in the analysis core, and Dr. Shastri in the development core. And we have collaborators in Shanghai to supply the co-infected patients, because in China it's a major health issue. 1/3 of population are infected. So we have plenty of samples over there. And the funding is from CFAR. And my own lab is funded by R1 grant that's on the basic biology of NKT cells. Thank you.
Questions? So can I start? So is there NKT cell memory, and then by stimulating them, are you stimulating durable NKT cell responses?
So the question is whether NKT cells have a memory. So it's a very good question that we have another basic project on that. So that is, after repeated stimulation, what is the transcription profile and epigenetic profiles of genes, and so you have to stimulate them in a right way, so you can have repeated activation. So if you stimulate without the co-stimulatory molecules, without IL-12, you are not going to have memory. You will have energy, that's called. That means after one stimulation, you are done. The NKT cells are not responsive anymore.
But with IL-12, you can generate a durable memory response.
With our protocol, that's the genetic cell loaded alpha galactosylceramide, and we know they produce IL-12. And in mouse models, by genetic knockout mice, we know that he is dependent on IL-12 and the CD40 ligand is the pathway.
Other questions?
So do the NKT cells have direct anti-mycobacteriocidal activity, or is it all mediated through CD8, CD4 B cell stimulation?
Yeah, so the question is whether the antiviral effect is dependent on the NK cell themselves or the downstream cells. So in the TB models, we know our TB model is dependent on CD4 cells. So we don't see any treatment effect. Actually, the mouse died within 2 weeks if you do the experiments in class 2 knockout mice. So we know in our model it's dependent on the crosstalk between NKT and the dendritic cells, then the CD4 help cells. And we believe it's a Th1 differentiation of the CD4 help cells.
So do you think that those cells could be used to differentiate iris versus progression of TB?
So whether these cells are responsible for the path to IIS or active TB?
Because what we see in clinic is, or in the clinical scenario, is a patient that has advanced HIV and TB disseminated, gets started on antiretrovirals, ATT as well, and it starts getting worse. And you don't know if you should start heavy steroids or that you should just continue with the ATT and assuming that that is iris. And you showed that these cells you were mentioning increase rapidly with antiretrovirals. So can you use them as a tool to differentiate these two very different conditions with different treatments in the clinic?
So we in the grant we proposed the aims to study the correlation. So our current hypothesis is if NKTs are more Th1 biased, if they produce Th1 cytokines, that should prevent the active disease. And whether it's correlated to IIS, that one we don't have a clear hypothesis yet, honestly. We have to see the results.
Okay. Let's think, I think.
Thanks.
So our next speaker will be David Murdoch from Duke University. He's going to speak on the longitudinal assessment of T-cell reconstitution in co-infected patients.
Thank you. Can everybody hear me okay? I aim to avoid the stop sign that she's holding there, which I think I can probably...
It doesn't turn off.
It doesn't turn off?
Yep.
Okay,
So
so be
not to
careful.
blind the audience.
All right. So for those of you I don't know, my name's David Murdoch. I'm from Duke, and there as part of the Duke CFAR. Been there for a few years now, and mainly involved in the flow cytometry core and doing immunology and flow cytometry work, and really sort of specializing in polychromatic flow cytometry panels. Presenting on behalf of my, co-PI on this gracious supplemental work award, Colin Menendez in South Africa with CHRU, although he's now out at Barra as well, and my colleague Melinda Suchard, who's in the University of Witwatersrand in the Department of Hematology there, where I should note that all of this work is being done. So this is an in-progress study, so I'm going to spend a little bit of time on the background to get you to understand a little bit why I propose this and why I'm interested in this. But as we know, absolute CD4 counts and viral load remain the cornerstone of assessing HIV infection and immune status. And they overall have been beneficial in terms of being able to stratify patients at risk for various opportunistic infections, and I don't have to belabor that point. But they remain, quantitative and measures of immunocompetency, and I think when we start talking about or wondering why someone with a CD4 count a little bit higher, when you see them on the wards, why they develop TB versus someone who, we know that less than 200, particularly less than 100, should, and we know a bit more about the immunology there, they kind of fail to help us in these higher CD4 counts and sort of really look at risk. So the assessment of qualitative immune function to co-infections such as TB are only inferred with these tests. And other measures like immune activation, which I've also dabbled in, are confounded by numerous other clinical factors, other co-infections, parasitic infections, for instance. So those more general measures of immune activation, although sometimes predictive, are easily confounded. So I wanted to, and those as mentioned by CD38 and HLA-DR. So measures of TB-specific immunity, I think, would help really identify patients with suboptimal responses to co-infection and help us better understand and stratify or even predict patients who are particularly at these higher CD4 counts when on the wards in particular are often frustrated by the inability to really tell, one, a good diagnosis, and two, why a patient who has a CD4 count higher is developing TB. And there's no doubt it's a very complicated picture, but this is at least our focused attempt on trying to help with this. And so as a little bit of background, this is a good 10 years ago now out of Cape Town. I don't have to tell you, extremely high incidence of TB. And this was by published about 10 years ago, and looking at just the incidence of TB in these different CD4 strata and the effect of antiretroviral therapy. So this shouldn't be surprising when you institute antiretroviral therapy, and I should say this was done in patients who were undergoing a lot of clinical trials at the time when antiretrovirals were available in those trials, and then compared to a cohort in the mid-90s who were not receiving drugs at that time. And they actually had a pretty good definition of TB, either by autopsy or culture, or AFB presence. But you can see what the effects across CD4 strata. But what's interested me is that you still have a heck of a lot of TB here in this 2 to 350 range. And so I really wanted to try to focus on this group here and try to understand what is it that's going on in terms of antiretroviral therapy and reconstitution, the normal immune reconstitution in some of these patients. So we do know that cytokines such as interferon gamma and TNF alpha are key, and they contribute significantly to TB immunity. There's also quite a bit of evidence with early Th1 polarized T-cell responses are important and occur as early as 20 days following TB infection in mice and other animal models. So clearly this balance is important for TB immunity. And polyfunctional effector T-cells appear to be important in a number of other areas, including vaccine-mediated responses, HIV progression, and some evidence although less so, in TB disease. And suppressive Tregs are also out there, of course, in the literature, and whether or not their role in the periphery and at sites of active TB may contribute to suppression of TB antigen-specific responses. So this is a little bit about something, about what we know. And in terms of reconstitution, we have small studies that have actually done longitudinally. This is by Wilkinson's out in Cape Town, and who looked at reconstitution over a number of weeks following antiretroviral therapy. And I should say these were all in patients with less than 200 CD4 counts. And in short, you have a CD4 positive interferon gamma positive and CD4 positive IL-2 decline in response to PPD over time, which is sort of represented by these sort of downward trending bar graphs here. But overall, there's a general expansion of central memory cells early, with naive expansion by late, which is some of what we've known. But what's interesting is that when they compare these patients who've undergone over a year's worth of antiretroviral reconstitution, they never returned to sort of normal subjects who were obtained from the same community. And if you look at the PPD responses, they, and this is by ELISpot, confirmed by their flow cytometry as well, you see that kind of peaks at week 12 and then declines, and this is thought to due to the later expansion of naive cells. So we came up with a number of aims. The aim number one being to quantify TB-specific T-cell functional immune responses in HIV-infected patients, and specifically looking at polyfunctional T-cell responses across CD4 strata, including those above 350 and up. We've also developed a polychromatic T-regulatory cell panel, flow cytometry panel, which actually phenotypes and reliably enumerates Tregs that we want to also apply to really get at this question of whether or not Tregs are playing a role here in T-cell reconstitution. And then simply aim 3 will be harvesting the supernatant and just looking at broad cytokines via multiplex assays. And the overarching study goal here would be to perform comprehensive T-cell phenotyping and functional assessment across CD4 strata. And I would hope through a better understanding of particularly these higher strata that we can better predict which patients are at TB and to also predict treatment responses and better know how to follow up and screen patients. Now whether or not any of this complex flow will be applicable at the bedside, remains to be seen, but it's possible. So this is a small investigative immunological study. We linked on recently with the CIPRA-SA study cohort repository, and I'll get to that in a little bit, but we're doing longitudinal phenotyping, 4 time points across CD4 strata, 20 patients in less than 220 in the higher, and then 10 above 350. These are very hard to get, as you can imagine, and so we have located those thus far. Earlier this year, when I went down and sort of started setting all this up, this is an example of sort of some of the patient specimen testing that we've done on other normals and TB subjects that we began to recruit. And this is an example of what we hope to exemplify in terms of polyfunctionality, where we can then do spice figures and look at these, and not only that, but across subsets, I should say, the subsets that were mentioned earlier. such as central memory, effector memory, and effector cells. And so we also have CD27 and 57 in the panel, and we can subset this out as well. And I hope to actually correlate this work with some of the other work I've been doing on specifically on iris and doing bronchoscopies and looking at the immune response within the lung compartment. And this is a very interesting patient because, of course, I'm going to show you the best one, that underwent bronchoscopy for suspected TB and suspected iris, who ended up actually having iris, but just technically after the 3-month definition window. But the top panel here is what their PPD stimulated responses were in the lung lavage fluid, and here's what they look like in the peripheral blood. And this always has highly interested me, and I've done about a dozen of these thus far, and have had about 2 or 3 patients that really mount this incredible local compartmental response. And I'd like to look deeper here and use some of the data that we're going to be generating via this pilot study to correlate with some of these other things that I'm seeing in other compartments. This is an example of the Treg panel that we have developed. We've applied this, I should say, across numerous HIV-infected patients, as well as tons of normal donors, and including other disease states, including other autoimmune diseases. And in short, we can, through a number of Treg-specific markers, do a lot of Treg subsetting here, and actually look within the phenotype here and actually reliably enumerate. So we're going to apply this as well going forward. This has been the project timeline thus far. As I was mentioned earlier, in the year began the work, and then the availability of actually lots of specimens became available through Ian Sana and CHRU and Robin Wood, who has completed this CIPRA assay study. So we're actually able to link on to another existing set of specimens, so we don't have to reinvent the wheel, and it's amazing that these were all drawn at the exact same time points we needed. There's literally hundreds of patients and thousands of samples, so we're able to actually go in now and actually pick these out and actually do batch testing and go forward and do them all at the same time. So you can understand that went through a couple months of IRB modifications and subcontract modifications and all that good stuff. But I'm flying down Sunday and next week going to start all of this and commence the batch samples. So they've all been transferred over. So we're in a sort of the throes of it right now, but I've shown you some of the preliminary stuff that I've done there on the ground. And I'd like to thank all the people involved, particularly Ian and Robin, and making this available to us, and my co-investigators there, largely supported by the Duke Flow Cytometry Core under Kent Weinhold and all of the wonderful staff there, and the other players here helping logistically, which it's all being done there, so it requires a bit of moving things around. So it's all been working thus far. So that's it. Set that like that. So any quick question? Yeah.
Yeah, just in terms of looking at localized responses other than in the lung, I was wondering if you, particularly with HIV co-infection, if there's been any other thought given to other tissues where you might look, and if you've done so.
Now I'm a pulmonologist, so I'm going to stick to the lung. But well, I should say we are dabbling with autopsy data that we have done systematically on patients who've